Hi, everyone. I'm Chase Weidmann, a new assistant professor at the University of Michigan. I just started a new RNA laboratory this past September in the biological chemistry department of the medical school as a member of the Center for RNA Biomedicine and the Rogel Cancer Center there. This is my first experience with the integrative RNA biology community through the ISMB, so I'm excited to be giving this presentation. Thanks to the organizers for inviting me. So the ultimate goal of my new laboratory is the identification of critical RNA-protein interaction networks that could become potential therapeutic targets. And our strategy for understanding these new targets will largely employ chemical probes in cell culture, freezing RNA-RNA, RNA-protein, and protein-protein interaction network information in place, and then using varying technologies to read that information out and rebuild what those native complexes looked like in cells. I'm trained as sort of a molecular biologist mostly, and my interest has slowly begun to require more and more bioinformatics. So today I'm going to be going to go through 3 short stories along this sort of ongoing journey, scaling up from studying repression mechanisms of a single RNA protein complex, trying to map new complexes on disease-associated non-coding RNAs through structure probing, to developing a new method to map protein interaction networks on RNAs directly, and then the future goal being to apply these technologies transcriptome-wide in an effort to be able to model RNA-protein interaction networks in cells from experimental chemical probing data. So I'll first sort of summarize the project that set me on this path. A big chunk of my graduate work was focused on an observation about the fly RNA binding protein called pamilio. So at early stages, fly embryos exist as this large syncytium where nuclear divisions are occurring in a shared bulk cytoplasm. The pamilio protein is expressed everywhere. However, it's exclusively regulating this hunchback mRNA target along a gradient towards the posterior end. And this is because a pamilio binding partner, nanos, is present as this gradient emanating from the posterior pole. And together, pamilio and nanos combine to repress the translation of hunchback along this gradient. And I spent a lot of time looking at the mechanism of this enhancement. Like native gel shift assays are a tried and true method to assess RNA protein binding. And many before me had used gel shift to measure pamilio's affinity for RNA. I just pushed it a little further, observing this trimeric pamilio nanos and RNA complex. Interestingly, I found that nanos can only interact with an RNA target as PUM is already bound, and it doesn't really have a capacity to bind RNA itself. But when nanos is bound to pamilio, the apparent affinity of pamilio for RNA increases 3-fold. Nanos seems to lock pamilio onto RNA targets. And with the help of Tracy Hall's lab, we got a crystal structure of this complex, which shows how nanos actually clamps pamilio onto RNA, binding both pamilio and upstream nucleotides. Now, I want to stress how small this interface is between less than a dozen amino acids on either protein and contacts with only a few nucleotides. And yet this small clamp enables pamilio to bind targets with really weak binding elements, targets that pamilio would not bind to on its own. And we also saw that a nanos-pamilio complex enriched an altered motif in vitro compared to what pamilio would pull out alone, shifting specificity from the 3' of the pamilio binding site to this 5' nanos clamp. And in crystal structures of the complex bound to weak RNA targets, you could even see a loss of resolution at a target's 3' end. There's a nucleotide missing here. And this is a consequence of its sort of relaxed recognition at that location. And this made sense too, as there are actually endogenous RNA targets with weak binding sites that are only repressed in regions where nanos protein is concentrated. And so this single protein-protein interaction shifted the potential amount of regulatory events from 25% of fly transcripts to 75% of fly transcripts, as these sort of combinations of RNA binding proteins could alter the fates of transcripts globally This is sort of what pushed me into thinking about using sort of sequencing and bioinformatics technologies to try and understand these networks rather than individual complexes. So this experience sort of informed my research vision going forward, right? I wanted to identify and characterize these dynamic RNP networks in cells. And at the time, the closest you could get was through these sort of cross-linking and immunoprecipitation or CLIP experiments. But this could only really evaluate one protein at a time, and it couldn't reveal how multiple proteins might interact with an RNA simultaneously. There was, however, this developing technology in the Weeks laboratory at UNC that could evaluate RNPs indirectly through measuring RNA structure. So for my second short story, I'll highlight some of the information you can get from these so-called shape map approaches. So shape and shape-like reagents, of which there are now many created and identified, they all report on RNA structure and some on other RNA properties. And I've helped direct multiple projects that rely on the shape reagent 5-NIA, which has a minute timescale half-life. It works pretty well in cells, and it reacts largely evenly with all 4 nucleotides, enabling easier analysis of its data. So shape reagents like 5-NIA selectively react with the 2' hydroxyl of flexible nucleotides, meaning that nucleotides in loops or single-stranded regions are moderately to highly reactive with shape reagents, while base-paired nucleotides are largely unreactive to shape. And that means that probing an RNA with a shape reagent essentially records that RNA's structural state, marking flexible nucleotides with chemical adducts. And using modified reverse transcription conditions, we can coax an RT enzyme to introduce mutations and deletions in product cDNA at the sites of shape reactivity. And then we just read that information out by sequencing. Mutation frequencies are converted into free energy change terms that restrain nearest neighbor folding parameters. As an example, here's shape data on the U1 RNA, whose structure is known. We see expected reactivity patterns here. So base paired stems are largely unreactive, so you see they're in black, whereas unpaired loops or single-stranded regions are moderately to highly reactive, or yellow and red. And even if we didn't know the structure ahead of time, like I said, shape data can be used to model base pairing. And depending on the number of potential pairings that fit the data, you can assign pairing probabilities to all putative helices. In this case, a single structure is highly favored by the experimental data, and that's why you see all these green high probability arcs here. And this does a pretty good job correctly identifying all the known structures of U1 in a minimum free energy model. You'll see it also models this extra helix in here, which likely only forms in the absence of proteins. And indeed, the U1 RNA is engaged by multiple proteins in cells to form a ribonucleoprotein complex. And shape data can actually report on these protein binding events as well. So you can see that this extra helix can't form if it's engaged by this SM protein complex. One thing to note here, if you look, is that actually most sort of single-stranded regions of this RNA are bound by proteins. So actually, most of this complex should be protected from shape reactivity if you probe it in cells. And that's exactly what you'll observe if you probe U1 RNA in cells. So here's sort of cell-free, protein-free shape probing versus in-cell shape probing. And you'll see that there's this big increase in the error at each nucleotide here. And that's actually a consequence of reactivity normalization on an RNA that now has become almost entirely unreactive. So I applied this sort of approach in cell and cell-free shape probing to try and understand multiple non-coding RNA protein interaction networks. And around the time I was developing that approach, I met Karen Schmidt at the time in the Novena lab at Dana-Farber. And she was working on this link RNA whose expression was tied to lower survivability of melanomas. So high expression of this Slinker-1 RNA meant a higher prevalence of metastatic disease. And she found that this Slinker-1 RNA was actually co-opting this protein BRN3A and the androgen receptor to activate expression of metalloproteinases, enhancing metastatic potential. She also saw that only a small fragment of this link RNA was necessary for metastatic potential. Deletion of just this one small region inhibits invasion through an extracellular matrix. And this is a necessary step for cancer cells to migrate from primary tumor to metastatic site. Ultimately, you actually only need this one region to promote that invasion. And the fragment size was actually perfect for investigation by ShapeMap, which is where I came in. So first, I extracted RNA from patient-derived melanoma cells and probed with 5-NIA. And when I looked at this region of Slinker-1 that was sufficient for metastatic activity, I found it was actually broadly reactive to SHAPE and isn't likely to be forming a lot of persistent structure. So as a comparison, look at this compared to U1 from the same cell line, which has a single stable structure. So you'll see well-defined structures have extended regions of these low reactivities at stems, and then these characteristic hyperreactivity at loops. And so if you squint at the Slinker data, maybe there's a single well-formed helix right here, but potentially not anywhere else. And overall, the structure of this region is much less well-defined, say, than the U1 RNA. Regions of low shape reactivity and low Shannon entropy are usually good indicators of stable structures. For example, the entirety of U1 passes these thresholds. Conversely, only a small portion of Slinker-1 passes this threshold. However, I did find significant protections from shape reactivity across this whole region when probed in cells, suggesting that maybe its inherent flexibility here and is necessary to recruit a number of protein complexes. And indeed, shape protections included a BRN3A binding site and 2 androgen receptor binding motifs. Now, by gel shift and SPR, we found that the androgen receptor N-terminus preferentially binds to unstructured pyrimidine-rich motifs, meaning that if this sequence were to be base paired, the androgen receptor won't bind to it. And this is sort of in agreement with our shape data that suggested that this region is highly flexible and unlikely to be extensively base paired. Thus, this flexible region, when highly expressed, brings the androgen receptor together with the BRN3A binding site, forming a non-canonical transcription complex that promotes metastasis. Because it is reliant on the absence of base pairing in that region, we found you can inhibit metastasis with oligos complementary to the androgen receptor binding motifs. You could also inhibit complex formation with single-stranded RNAs that mimic the androgen receptor binding site as well. Now, I bring this up as an example of how the sort of lack of RNA structure, not the presence of structure, at least base paired structure, drives the disease phenotype of this RNA. But not all long non-coding RNAs behave this way, as these sort of floppy protein binding sponges. So here's the monk long non-coding RNA, which controls differentiation during myogenesis. And you'll see the monk RNA appears to fold into multiple distinct stable structures. You see a lot more motifs here that resemble the U1 RNA and has pretty well-defined character throughout. And some of these regions were actually protected from shape reactivity in cells as well. In particular, the CH4 motif bore multiple protected nucleotides. Now, my collaborator at the time in the Dutta lab, Ruja, had a number of established assays in mouse myoblasts that measure pro-myogenic function and was able to test how deletion or mutation of each of these regions affected phenotypic outcomes. And she found that full activation of the MyoD1 gene by monk required pretty much every motif to be intact and was especially dependent upon this CH1 and CH4 motifs. What was really interesting is that different regulatory targets required different combinations of the structural motifs. Activation of two other myogenic targets, MyoG and MyH3, did not depend on all of the motifs. And MyoG could even be activated without the usually critical CH4 domain. You also don't see that mutant complexes are able to rescue each other in trans. So CH1 and CH4 have to be on the same RNA molecule in order to promote a response at targets that depend on them. So in all likelihood, these domains are intentionally bridging components together in a distinct complex. One question with these kinds of experiments is always whether an identified structure that we see is actually important, or is it just dependent upon the underlying sequence? And so to test this, Ruja made a number of mutations in the critical MUNC motifs using transition mutations that conserve base pairs and retain purine pyrimidine character while scrambling the exact base identities. And for most regulatory targets, none of the mutants behaved significantly different than the wild type sequence. Only a deletion of the whole structure could block any activity, suggesting that the activity depends on the presence of the structure motif in some form, but not the exact sequence. Activation of MyoG was dependent on this loop sequence, though, suggesting that some activation complexes do recognize specific sequences as well, but it's specific to the target. The other critical motif, CH4, was almost completely unaffected by these similar transition mutations that conserve structure but just change the sequence. So you can see by structure probing, you can identify important functional motifs, but you won't know whether a flexible region or a structured region is more important than the other. Slinker and MUNC sort of present these opposite extremes. A short, unpaired, flexible region in Slinker bridges a transcription complex together, while this network of MUNC structured motifs work together to fine-tune expression at an assortment of different pro-myogenic genes. But what was true in both cases, again, was that the important regions were being engaged by RNA binding proteins. So I've taken you through another step on my journey here, using chemical probing of RNA structure to try and characterize some link RNA mechanisms. But I still wanted a better way to measure protein interaction networks on RNA, because shape is really just providing an indirect measurement of protein interactions by reporting on these in-cell protections. I wanted a probe that physically reacted at sites of protein binding on RNA, and ideally one that could report on protein-protein communication across an RNA, which leads into the last story I'll talk about, this new technology and data type I've developed and have been enhancing called RNP-map. Ultimately, I identified this chemical probe, SDA, which has an amine reactive group and a photoactivatable group. The amine reactive group selectively reacts with flexible free amines, like those in lysine side chains, which are one of the most prevalent side chains in RNA binding domains. Upon photoactivation with long wavelength UV, the diazurine group can rapidly cross-link labeled lysines to nucleotides in close proximity. SDA is cell permeable and thus can be used to cross-link RNA protein complexes in live cells. Cell lysis and protein digestion then produces RNAs with short peptide adducts. And just like with shape modifications, I use altered reverse transcription to encode the location of protein binding sites as mutations in product cDNA. Because these adducts are being read through instead of generating termination events, we can also see multiple cross-linking events on the single RNA molecules. So here, nucleotides with high mutation frequencies represent sites of protein binding. And we also observe nucleotides whose mutation statuses are linked, either being significantly co-mutated together or always mutated alone or individually. These events could result from a protein-protein interaction across an RNA, the same protein bridging two locations together, or a coordinated assembly event. And with combinations of these correlated nucleotides, we can begin to describe complex protein interaction networks on RNA. And so I called this RNA protein networks analyzed by mutational profiling, or RNP-map. And I find that nucleotides with the highest SDA reactivity correspond to known binding sites very well. For example, I identified every major binding site on this U1 RNA-protein complex. And reactive nucleotides I found are proximal to protein lysines, as expected, because that's where SDA should be reacting. We also get single molecule information, correlations between protein-bound sites, including a known long-distance protein-protein interaction between the 70K protein and the SM protein complex. We also see a bunch of correlations that emanate from sites that bind the SM complex, which is responsible for initiating U1 RNP assembly. Now, the ideal would be if these RNP map profiles could predict what these functional assemblies look like and even identify the proteins involved at the same time. Well, we're not there yet, but I do think there is a number of identifying information within these RNP map profiles. So as another example, RNase-P and RMRP RNAs are structurally conserved but sequence divergent. These are RNAs that form RNP endonucleases. While they have pretty different sequences, their shared structure enables them to bind largely the same set of proteins. And as you might expect, for two RNPs that should look the same structurally and bind the same proteins, the RNP map site patterns are thus very similar throughout both RNAs, with patterns that mirror each other. And the region where they vary the most is in their specificity domains, which makes sense because these domains are what allow them to target different substrates. So that means buried in the structure and protein binding information are the features that define this RNP. And this is even more evident in their correlation profiles. So if I align RNase-P and RMRP based on their shared structural motifs, I find that their protein interaction networks are nearly identical, even though these RNA sequences aren't strongly conserved and they differ in length by 80 nucleotides. And if we look at where these shared networks are, I find that these RNPs are defined by protein interaction network hubs, where multiple strong correlations link together specific regions. And these shared network hubs actually define the substrate active site, the specificity domain, and the protein components that bridge the specificity domain and active site together. So essentially, in one RNP map experiment, you can highlight important functional regions of an RNA of interest, irrespective of sequence conservation. And so if we can extract more of the useful information from these profiles, we could potentially identify unique functional RNP domains transcriptome-wide. But first, again, there's still some rules we have to figure out about this new kind of data. For example, say, what about proteins whose binding sites don't rely on lysines, like, say, Pamilio? I actually would have been very upset if the RBP that led me down this path couldn't be captured by this method I was designing. So here's a 2.6 angstrom structure of human Pamilio, PUM1. Modular repeats in this RNA binding domain specifically recognize and stack with each base in a specific RNA binding site. And indeed, if you look at this, there's no lysines involved. And at first I was worried that some interactions like this might be missed in this procedure. But while there aren't lysines directly recognizing the bases, they do line the outside of the binding domain. And this is true for lots of RNA binding domains as well. And this includes forming this sort of cage around the 5' end of an RNA target. And in reality, every base, although not directly involved with a lysine for binding, every base in the binding site is within about 10 angstroms of one of these lysines. And I do indeed see signal at PUM1 RNA targets. For example, here I've labeled the SDA reactive nucleotides in the P27 mRNA 3' UTR. Now, expectedly, the caged 5' end site is highly SDA reactive. Interestingly, the other reactive nucleotides include this central region right here, which are actually the furthest from lysines. Now, these positions are known to be more weakly oriented in the structure and are actually even flipped out in some conformations. So maybe there's some inherent flexibility that increases their reactivity in this assay. But there's clearly additional information about RNA-RBD interactions within these RNP map profiles. Now, Towards being able to decipher some of this information buried in the RNP map profiles, I've mostly been working on improvements to the signal from the experimental end. So while I was developing RNP map, a group at the University of Cambridge came up with a simplified strategy to enrich cross-linked RNP complexes with trizol, and they called it orthogonal organic phase separation, or OOPS. And I wondered if this enrichment could enhance RNP map signal. And indeed, comparing enriched cross-linked fractions to free RNA fractions from OOPS, this increases the signal about threefold. And funnily enough, of course, it actually makes the experiment easier and requires half of the cellular input material. So that was a great find. The enrichment process doesn't favor any kind of signal over another. Signal at all nucleotides is enhanced, as well as all types of mutation events as well. So one issue in the original approach is we'd have to throw out a whole bunch of these single molecule correlations, because there's actually very little difference in the distributions of background co-mutations and treated sample co-mutations. We'd find real events, but you had to sort of dig deep with lots of sequencing power. Now, with the added enrichment step, the signal to noise is dramatically increased. And this actually translates to richer interaction networks from smaller data sets. And this makes transcriptome level data much more approachable going forward. We had also originally had to use these empirically determined thresholds from known RNP structures, of which there's actually only a few complete ones with atomic resolution data. But this increased signal with OOPS enabled us to be more confident in our binding sites without relying on potentially biased small data sets. Now we use a random sample consensus approach to find a background model within each new data set we collect, and then identify outliers with specified confidence depending on the strength of that model. And because there's some bias and enhanced reactivity between each nucleotide, we do this separately for each. Now, importantly, this new analysis, and especially the OOPS step, enhances our ability to find nucleotides within close proximity to lysines. And the hope is that we'll be able to use this enhanced binding profile information to discover RNP elements and motifs transcriptome-wide. So to summarize, I told you about the RBP-RBP interaction that got me interested in looking at RNA-protein networks. I showed you some structure probing approaches to understand lincRNA function, and I told you about a new technology and data type, RNP map profiling. And my hope is to apply these new technologies transcriptome-wide and to couple them to cross-linking based proteomics approaches with a major focus on non-coding RNA protein complexes that drive mechanisms of cancer metastasis. So with that, I'll acknowledge my new faculty home, along with the funding and collaborators that made all the work that I showed today possible.